Hey everybody, my name is Sarah Waldo. I'm a graduate student in the Lab for Atmospheric Research here at WSU, working with REACCH in the second objective of measurements and monitoring. And I'm going to talk about my work measuring nitrous oxide emissions from agricultural fields using a combination of micrometeorological methods and static chambers. You may be familiar with nitrous oxide because it's more commonly known as laughing gas. It's not the same thing as this cousin Larry in this cartoon here. Luckily, that's not what I measure. But in addition to having some medical uses, it's also a greenhouse gas that has 310 times the warming potential of carbon dioxide on a molecule per molecule basis. And it's a long-lived gas. It has a lifetime of about 100 years, which means that it gets mixed into the stratosphere where it destroys the good ozone up there. So for these reasons, people who study nitrous oxide like to say that it's no laughing matter. I also wanted to take a moment to point out that nitrous oxide, or N2O, is in a different chemical family than NOx, which includes NO and NO2. And this has to do with the kind of reactions it undergoes, the lifetime. And so it's just important to remember that N2O is a greenhouse gas and ozone depleting substance. NOx is a tropospheric pollutant. So where does this nitrous oxide come from? Its main source is that it's produced as a byproduct of the microbial processes in the soil of nitrification and denitrification, which are shown here. Okay, so N2O is important in agricultural systems because agricultural soils, which are fertilized, produce a lot more nitrous oxide than natural soils. So this pie chart shows the amount of global warming, the amount of warming that's caused by nitrous oxide compared to other important greenhouse gases. And you can see that CO2 is most of it, and then methane is the second most important, and N2O only makes up about 6% of the total warming. But we're really interested in it in agriculture because agriculture is such a big source of N2O. Agricultural soil management, as you can see from this second pie chart, accounts for about 3/4 of the source of N2O. And agricultural soils are a bigger source of N2O than natural soils because of all the fertilizer that's added to them, all the nitrogen fertilizer. So in the Pacific Northwest, in wheat-based cropping systems, for a winter wheat crop, generally around 150 to 200 kilograms of nitrogen are applied per hectare. And only approximately half of that's used by the crop, and I'm nervous right now for what Dave Huggins is thinking while I'm talking about this. Hope it's about right, Dave. Only a few percent of the nitrogen applied, only a small amount of it is lost as N2O. And so it's not as important from a nitrogen budgeting aspect. You're not like, it is an efficiency issue, but it's not like the farmer is losing a ton of money in the form of nitrous oxide, but it's important because of its environmental impacts. So the IPCC currently has a rule of thumb estimate of 1% of the nitrogen applied will be lost as N2O, but we really are interested in seeing what that number is for this region. So the reason why this type of measurement hasn't been undertaken before is because nitrous oxide is a difficult gas to measure. This is for two main reasons. The first is its variability, both spatially and temporally. There can be hot moments of N2O emissions due to soil disturbance like fertilization or tillage, precipitation, or freeze-thaw cycles. And then there can be a lot of spatial variability or hot spots because of soil properties like soil temperature and moisture, soil type, and also having to do with the microbial community in the soil. And then the second challenge has to do with the detection limit, or our ability to detect small changes in concentrations of nitrous oxide. So the way we're measuring nitrous oxide is by combining chambers and a tower-based micrometeorological method to measure both the on a fine time scale as well as a large spatial scale. So we have these two sites out at Cook Farm, well, one on Cook Farm and one on WSU land across the way. One's a no-till site and one's operated under conventional tillage. And these sites that we're measuring N20 are co-located with the flux towers that are using the eddy covariance method, which you may have heard about before from people in my group, to measure fluxes of carbon dioxide and water vapor. We have 16 chambers at each site. They're Lycor automated static chambers. And the advantages of these is that they can measure really small fluxes of nitrous oxide, and they have continuous temporal coverage. A drawback to them is that they're basically, they have an area less than a square meter, so they're basically a point measurement of nitrous oxide. So there can be really high variability between the different chamber measurements. So this is why we're pairing it with the flux tower, using the flux gradient method to measure fluxes of nitrous oxide at the field scale. And this scales up spatially from the chambers, but it has some drawbacks in terms of temporal coverage. The chambers have been operating out at the Cook Farm no-till site since last spring, and we got the flux gradient system running there a few weeks ago, and then there's another system out at the Clark site that's been running since the beginning of August. This slide shows some preliminary results from the no-till site, looking at the 16 different chambers. You may have noticed in the last slide that the 16 chambers are broken down into 4 groups of 4. So we're going to go through and look at each of these groups individually. The theory being that there will be higher emissions of N20 from the chambers that are further downslope and have higher soil moisture. So this is group 4, the furthest downhill group, and you can see some diurnal variation in the time series. This time series showing about 2 weeks of results, and the y-axis is showing the nitrous oxide flux in units of nanomoles per meter squared per second. And we can see also that the average emissions are decreasing over time, and this kind of makes sense because there was a rainfall event around August 16th. So the rainfall event would spur emissions and then they'd peter off again. Now if we add group 2, which was the furthest uphill group, we can see emissions of about the same magnitude, and but we don't see the same decrease in emissions over time. If we look at group 1, which is just uphill of the tower, we see a more pronounced, one chamber has elevated emissions and a more pronounced decrease over time. And then if we add in group 3, we see this one kind of crazy outlier, but it also does have this diurnal pattern and the decrease in emissions over time. So this slide is to illustrate just how much variability there can be in the same field, even amid chambers that are in the same group of soil type and soil moisture. So this really illustrates why we need another technique in order to scale up to the field scale. So the bottom plot on this slide is showing that same time series of the different chambers grouped by color. And the black trace you'll see there is the average of all 16 chambers over the 2-week period. And then the top plot is showing that average in black and the standard deviation in the gray area, gray shaded area. And then in the red markers are showing the flux gradient measurements from each day, the average and the standard deviation. Also plotted on the top plot are the air temperature and the precipitation during this 2-week period. So this is really interesting because the flux gradient results imply that the outlier chamber is representative of the greater field. That outlier chamber brought the average up, and the flux gradient field integrated measurements are showing that that is more characteristic of the field scale. What we're trying to measure is the total N20, like what the field is, how big of a source of N20 the field is. And previous studies comparing chambers and towers have shown that they give similar results if the footprint's the same. But if we're interested in the whole field, we need to sort of figure out how detailed we want to be in terms of, you know, what's a characteristic footprint. But that kind of brings us to our future / ongoing. These chambers are out there measuring nitrous oxide as we speak. So these two sites with the paired chamber gradient technique set up are the first continuous long-term multi-site measurements for this area. So we're really interested to see just how the nitrous oxide emissions compare to other model estimates, the IPCC Tier 1 guidelines, and other measurements from other wheat cropping systems. We're interested in what the total N2O losses are on an annual basis to see what kind of source strength these fields have. We're really interested in characterizing nitrous oxide emission behavior, how emissions respond to soil temperature and moisture, freeze-thaw events. The plot that's in this slide illustrates a surprise we've already monitored for this project. This is showing chamber results from last winter, and we weren't expecting to see much nitrous oxide emission at all when it's cold out. The microbes aren't very active, but there was this freeze-thaw cycle in January that had some really high emissions. The emissions on the last slide didn't go any higher than 4 nanomoles per meter squared per second, and you can see here they're up to 14, 15 in some cases. We're interested in comparing sites to see if that sheds any light on management practices between conventional till and no-till. And we'll also be working closely with Claudio Stockel using CropSys to maximize the applicability of these results. Thanks, and I look forward to talking to you guys about this on Thursday.